So hello everyone, let us start the session for today. I hope my screen is visible. So welcome everyone to the week 5 PMRF-NPTEL session on the NPTEL course, the Introduction to Developmental Biology. The PMRF-NPTEL sessions are designed to be interactive problem-solving sessions in which a PMRF fellow such as myself interacts with the various students who are enrolled in the NPTEL course, and we together solve various assignment problems from the previous year's run of the course. These sessions are designed so that the students get better idea how to approach the assignment problems and also clear various concepts that were taught throughout the week. So let me first introduce myself. My name is Anuvab Chatterjee. I am a PMRF fellow and PhD scholar under the supervision of Professor Jonaki Sen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. This particular week in the course, there are various lectures on morphogenesis and cell-cell communication, and we are going to solve various problem questions based on these concepts that were covered. So to start off, let us go through the first question. The first question is, which of the following processes are regulated in a multicellular organism in order to ensure correct morphogenesis? The options are aggregation of desired cell types and their positioning, relative to each other within a tissue, cell migration to their desired positions in tissues, growth of organs as their cells grow, and formation of an organ to attain specific polarity. So this is a multiple choice question. So there are more than one option correct, and I would encourage everyone who are here in the meeting to try and answer. Deepak says the options are 1 and 2. Aggregation of desired cell types and their position relative to each other within the tissue, and cell migration to their desired positions in the tissue. Grinalini says all 4 of them. The morphogenesis also includes growth of organs as their cells grow and formation of an organ to attain specific polarity. So let us go through the solution slide. So in morphogenesis, the main questions that we try to answer include how tissues are formed from population of cells. So beginning, we all know that the entire organism always evolves from a single cell, which is known as zygote. The entire question is, how does the zygote, which then subsequently forms many cells, form various complex structures? This entire process of forming various structures such as organs, tubules, blood vessels from normal cells, which are essentially a plain sheet of cells, is what morphogenesis entails. Thus, the big questions of morphogenesis include how are tissues formed from population of cells, how are organs constructed from tissues, how do organs form at the right location. So this is another question. So say the location of our heart or our brain or our lungs are perfectly predefined and is consistent over the entire population. How does our body know that exactly in this location should a particular organ form? Or how do cells migrate to right location? Say we have already seen the example of how during gastrulation the various germ layers are formed by specific cell migrations, and how the neural crest cells migrate to various positions of our craniofacial areas in order to form the entire craniofacial apparatus and various pigment producing cells. So how do cells know where to migrate, when to migrate, and where to stop? These are all answers that questions that are asked by morphogenesis. There is also the question of how do organs and their cells grow. When particular organs are growing in size, specific cell population are also growing in size, but disproportionate growth of various cell population will make a non-functional organ. Everything needs to be orchestrated at a minute level, and this is also what falls under the category of morphogenesis. And finally, how do organs achieve polarity? So, as we have discussed, I think 2 classes back, that there are various axes that are designed that are used to describe various organs and positions of our body. These are anterior, posterior, dorsoventral, and medial and distal axes. Essentially, the X, Y, and Z axes that we use. So a particular organ always grows in a particular direction. It is not similar in all directions. Say our brain. Obviously, you know that the cerebellum is towards the front side, and then comes the midbrain, then the medulla oblongata, cerebellum part. So the entire organ has polarity. That is, it is different in different directions. And how is that achieved? This also falls under the prospect of morphogenesis. So to answer our question, which of the following processes are regulated in a multicellular organism to ensure correct morphogenesis? All of the options are correct. That is, aggregation of desired cell types and their positioning related to each other in the tissue, cell migration to desired positions, growth of cells as their cells grow, growth of organs as their cells grow, and formation of an organ to attain a specific polarity. Anyone has any questions regarding this? I guess. So we move on to another topic that was discussed. In the context of sorting of different types of cells in early embryos, the term selective affinity refers to, the options are migration of cells based on the surface tension, migration of cells to a final position that reflects their embryonic positions, sorting of mesodermal cells inside with endodermal cells outside when the 2 types are mixed, presence of a hierarchy of interaction among different cell types. So let me give a context to this particular question. So when various experiments were done during early days, where various germ layers were taken and mixed together to form a varied cell population, which is not normally seen in the normal embryo, everyone observed that particular cells started migrating in a specific manner, and specific population of cells tried to clump together while other ones tried to be separate from each other. Say that we had 2 types of cells. Cell type A clumped together while cell type B always tried to go away from the interior of the entire population. So this is what was known as cell sorting. So cell sorting was observed, but there were particular experiments that followed up, and there was this term selective affinity which defined something specific in this type of cell sorting. The question asks us, what is during sorting of cells, what exactly this selective affinity mean? The options, as I said, are migration of cells based on surface tension, migration of cells final position that reflect embryonic positions, sorting of mesodermal and endodermal cells outside when these 2 are mixed, and presence of a hierarchy of interactions between different cell types. So this is a round type of question, so only one of them is correct. I would like everyone to try and answer this. What does selective affinity mean? Deepak and Milanini say that it is number 2, migration of cells to a final position that reflects their embryonic positions. Anyone else wants to try? Srividya also says number 2, migration of cells to a final position that reflects their embryonic position. Okay. So let us look directly into the various experiments that were done. These experiments were the differential cell affinity experiments done by Townes and Holfenter. In these, various cell population, as I said, were mixed. So when epidermis and mesodermal cells were mixed, it was observed that the mesoderm always tried to, okay, Sneha says number 1, migration of cells based on the surface tension. Okay. So when various cell types were mixed, say epidermis and mesoderm, it was observed that mesodermal cells always tried to go inside while the epidermis covered everything. Similarly, with mesoderm and endoderm, there was specific cell sorting with mesoderm coming inside, endoderm outside. When epidermis, mesoderm, and endoderm were mixed together, it was observed the mesoderm was inside with the endoderm while the epidermis covered the entire cell population. When the neural plate and the epidermis were mixed together, the neural plate was inside, the epidermis outside. And finally, when the neural plate, axial mesoderm, and epidermis were mixed together, the neural plate was inside, neural plate cells were migrated inside, mesoderm, after which the mesoderm covered the neural plate cells, and finally the epidermis covered the entire cell population. Thus, we observe that sorting out and reconstruction of spatial relationships in aggregates of embryonic amphibian cells. That is, there was specialized spatial sorting of various cell types. So, depending what kind of cell type it was, it always wanted to clump together with its own cells and in a specific hierarchy with respect to the entire cell population. Final position after sorting reflects embryonic positions. Reaggregation of amphibian embryonic cells were done, and this entire sorting process, in which the final position after sorting reflects the embryonic position, is known as selective affinity. So I hope this is clear. Yes, what exactly selective affinity is? I guess. But I do have a question here. Can anyone tell me what is the difference between epidermis? Like, there are 2 or 3 terms that here are very confusing and need to be sorted out. There is ectoderm, mesoderm, endoderm, and then there is epidermis and mesenchyme. Epidermis and ectoderm and mesenchyme and mesoderm are very easily confused. Can anyone tell me what the differences between these are? Also, there is also the another cell type, epithelial to mesenchymal transition. Epithelial and mesenchyme and ectoderm and mesoderm are very confusing, and many of us often get it wrong. So, anyone wants to tell me what the difference between epithelial, mesenchymal, ectoderm, endoderm, and mesoderm is? Typing it in the chat box will also do. So no one wants to try, or no one has an idea? Could be either. Neha says ectoderm, mesoderm, and endoderm are germ layers, and Ujjwal says epidermis is formed from the ectoderm, which is absolutely correct. But the first ectoderm, mesoderm, and endoderm are germ layers, and epidermis is also from the ectoderm. But then, can you tell me what exactly epithelial cells are and mesenchymal cells are, and what are the origin? Anyone wants to try? Okay, so what Sneha says is absolutely correct. What Ujjwal also says is absolutely correct. Ectoderm, mesoderm, and endoderm are 3 separate germ layers. That is it. Period. We stop there. Those 3 need not be confused with any other cell type: epidermis, epithelial, mesenchymal. What happens is ectoderm gives rise to 2 separate cell types of cells, which are the epidermis, which forms our skin or the underlying layer of our skin, and the neural plate cells or the CNS, the central nervous system, is formed from ectoderm. Next, epithelial cells, a particular cell population which have a specific morphology and are present in layers with specific cell adhesion junctions which connect them to other epithelial cells. These cells form layers, are very organized, and form cell adhesion junction with every other cells. Mesenchymal cells are cells which do not have a specified morphology, can exhibit cell migration by themselves, and do not have cell adherence junctions with other cells. That is, they can roam around on their own. There is a specific phenomenon known as epithelial to mesenchymal transition that we'll go through later on, in which various epithelial cells might convert to mesenchymal cells. This is something that we'll go through later on in the lecture series. But this is something that I wanted to be clear about from the get-go, that ectoderm, mesoderm, endoderm are germ layers. They are completely, absolutely separate from epithelial cells, mesenchymal cells, which are types of cells. Epidermis is the part of the part of cells which rise from the ectoderm, which gives rise to our skin layer or underlying skin layer. Is that absolutely clear? The distinction between all of them, because these terms get more and more jumbled up as more further we discuss, and it is very easy to get confused. Is it absolutely clear what the all the terms mean? Yes, no, maybe. Okay. So with that, we try to answer our particular question, which was, what does selective affinity mean? Selective affinity means migration of cells to a final position that reflects their embryonic positions. Anyone has anything to add to this question?
So I have a doubt. Can you please explain that mesenchymal, what you were saying? I cannot
You want to want?
mesenchymal.
Okay,
That mesenchymal. Yeah. Yes.
So, during development, there is a specific cell population known as mesenchymal cells. Mesenchymal cells are cells whose morphology is not very well defined, do not form adherence junctions, and can migrate on their own. These cells are present in various developmental stages and later on get specified to a specific cell population. Epithelial cells, on the other hand, are cells which form layers of cells, are have very well defined morphology, and get adhered to various other epithelial cells to form those layers of cells. Mesenchymal cells, one of the examples of mesenchymal cells can be, obviously I do not have the diagram here. It is a layer of cells that covers our forebrain when it is developing, and this they migrate to specific various regions of the our forebrain and craniofacial layers to form various structures such as our nose, our jaws, our face. These are all found by mesenchymal cells, which migrate to a specific location and then differentiate. Okay, so we move on. It is in the We go on to the next question. How does surface tension or ST influence cell sorting? The options are: cells with different surface tension migrate such that the cells with the highest surface tension form the outermost layer and the lowest surface tension form the innermost layer. Cells with different surface tension migrate such that cells with the highest surface tension form the innermost layer and the lowest surface tension form the outermost layer. Surface tension does not influence sorting of cells. And finally, differential surface tension among different cell types ensures sorting that reflects their embryonic positions. This is again a multiple choice question, so there are more than one answer that are correct. Deepak says 2 and 4. Cells with different surface tension migrate such that cells with highest surface tension form the innermost layer and lowest surface tension from the outermost layer, and differential cell sorting in order to reflect embryonic positions. Mrinalini, Sneha and Ujjwal also agree with what Deepak said. Okay. So we go ahead and look at the slide. This was a particular experiment done by 40, in which what they did is they took various cell populations and calculated the surface tension of the cell population, where they formed individual cell aggregates. So when limbered green cells were allowed to form aggregates, they had a surface tension of 20.1. Similarly, pigmented epithelium, when they were allowed to aggregate, they had their cell surface had the surface tension of 12.6. But when both of them were mixed, it was seen that the cell type with higher surface tension was always inside, and the cell type with the lower surface tension was always outside. So this was seen in here, limbed and green cells. We see the green cells go inside, the red one outside. Similarly, the red and yellow cells, red goes inside, yellow outside, and similar phenomena is seen. Thus, cells sort to form aggregates with the smallest interfacial free energy. So this essentially follows thermodynamics and our knowledge of how entire everything around us tries to attain the lowest potential energy. Thus, all cell aggregates also try to lower their surface energy, and since surface tension is directly related to surface energy, we are able to see that the cell population whose surface tension is the highest tries to go inside the cell aggregate, while the cell population with lower surface tension is always on the outside. Thus, according to the question, when surface tension influences cell sorting, the highest surface tension cell population forms the innermost layer, lower surface tension forms the outermost layer, and differential surface tension among the different cell types ensures sorting that reflects embryonic positions. So these two options are correct. We are okay with that? Okay. I guess that was a yes. Right? So we move on. The selectivity in selective affinity arises due to, that is, why do given cell types prefer adhering to some cell types but not all. So as we have just seen, mesenchymal cells when mixed with epidermal cells, always mesenchyme comes on the inside, and both are completely separate to each other. So cells of one type try to adhere with themselves, while cells of another type try to adhere with themselves. So no cross interaction is there. So why is that? The options are: the difference in interfacial energy of different configurations of cell aggregates. Cells prefer configuration with the smallest interfacial free energy. The difference in experimental setup. Depending on the experimental conditions, cells may adhere differently. The difference in staining method. Two particular stains always result in a definitive configuration. When red and green cells are mixed, red cells are always localized in the center and green always on the surface as observed by 40. And the another option is both A and C. So this is single type correct answer. Okay, the options we are getting are: Mrilani says A, Sneha also say, Deepak says 4, that is both A and C, and Sneha and Uchwal also say A. So, and Srividya also says A. So depending on just the discussion we had, the option is A, that cells prefer configuration with the smallest interfacial energy. What the C option says is the difference in staining methods. Two particular stains always result in a definitive configuration. That is, the stain determines how the cells will sort, which is obviously not the case as we have just seen here. So when red and green are mixed, green is inside, red is outside. That does not mean if I stain limbed cells with red and pigmented epithelium with green, the configuration will change such that the green is still on the inside and red is outside. The stain just stains the cells, gives us what cells these are. The stain does not dictate how the aggregation will occur. So whatever the color of the stain we apply on the limbed cells, they will always go inside. Whatever the color of stain we apply on the pigmented epithelium, they will also always go outside when the two of them are mixed, irrelevant of which one is red, which one is green. What determines the sorting is the free energy, the interfacial free energy that is present on the surface of the entire cell aggregate. Is that okay, Deepak? Okay. Anyone else has something to add on this question? Okay. So we move on. The next question is, what is the role of catenins in cell adhesion? Catenins regulate cell-cell adhesion by binding to calcium. Catenins stabilize cell-cell adhesion by interacting with the actin cytoskeleton. Catenins stabilize cell-cell adhesion by interacting with microtubules. Catenins regulate cell-cell adhesion through phosphorylation. This is a single option correct answer. Mriani says B. Catenins stabilize cell-cell adhesion by interacting with actin cytoskeleton. Deepak, Shriyansh, and Sneha also say B. Okay. So let us look at the. Anyone else wants to try? Okay. So let us look at the solution slide. So there are a particular class of proteins that help in cell-cell adhesion. So we have learned, like we have had this discussion of how surface tension is important, how particular cells sort when they are with amongst similar cell types. But what proteins or molecules are responsible for this differential cell adhesion? These molecules or proteins are known as cadherins, or the calcium binding adhering proteins, or the calcium dependent adherent proteins. So these proteins look something like this. So these have an adhesion recognition site and a calcium binding site. Cadherins, when there are cadherins of similar types on close by cells, this adhesion recognition site is actually able to connect the two proteins. So there is cadherin binding that occurs, and this is what gives rise to our cell-cell interactions. Catenins, catenins, catenins are protein that interact with these cadherin proteins, and catenins are responsible for linking the cadherins to the actin cytoskeleton of the cell. So here we can see a similar an experiment done in which a oocyte is injected with an antisense oligonucleotide against a maternally inherited cadherin mRNA. The inner cells of the resulting embryo disperse when the animal cap is removed. So when the outer animal cap is cut open and cadherins are inactivated inside, then we see that all the cells essentially spill over, that there is not much cell-cell adhesion that there is present. But in the wild type embryo, we can see that all the cells are adhering together. So cadherins are responsible for the cell-cell interactions. Catenins, on the other hand, are responsible for interaction between the cadherins and the actin cytoskeleton. Thus, the role of catenins in cell adhesion is catenins stabilize cell-cell adhesion by interacting with actin cytoskeleton. Is that okay?
So I have a doubt from this slide.
Yes.
So in this slide it is said the oligonucleotide was produced against maternally inherited cadherin mRNA.
Yes.
Then does it mean that the cadherin is having some sort of maternal heredity only?
That is a good question. So during during primary stages of development, as we will see in Drosophila embryo oocyte development, you will see that specific mRNA and specific proteins are already inherited in a differential polarized manner. So before we a certain cell division stage, the translational apparatus of the zygote is not yet active. So many a times what happens is the maternal mRNA and the maternal proteins deposited already inside the oocyte, not the zygote, already in the oocyte, dictates specific developmental functions. So for example, if you know about the becoid and the caudal mRNAs, which help in determining the anterior posterior polarity of the Drosophila embryo, these are already inherited by the oocyte from the maternal these are maternal mRNAs and proteins. These are not zygotic proteins. So the in the starting stages of development, these proteins are what play major roles in development. Here, that might be the case that the maternal cadherin mRNA is playing the major role in development, and thus when that is inactivated, we see this disintegration. It might also be possible that since the since it is not said which cadherin was inactivated, it might be the maternal cadherin plays a major role in this particular stage in interacting, and thus when we essentially knock it down using antisense oligonucleotide, we see such an effect. It might be either of them. Is that okay?
Yes. Yes.
Okay. So I have a follow-up question on this. On this particular site, there are a number of proteins that are shown. Is a particular protein popping out to you to be related to a specific a specific signaling pathway? Do we know a particular protein that is the direct readout of a particular signaling pathway? And we know that there are not a lot of signaling pathways. This is something that was not discussed in these lectures. Like we'll probably discuss in next week. There are the RTK pathway, JAK-STAT pathway, Wnt signaling, BMP signaling, FGF signaling, and RA signaling. These are essentially what we have in our hand. Do we know any signaling pathway which has one of these proteins shown here as output? Yes, Kunal, do you want to try and answer that? Okay, so what I wanted to say is one of the proteins shown here is a direct readout of a particular signaling pathway. So we know if the signaling pathway is active, maybe it is directly controlling this particular protein. Can anyone guess which protein I am talking about? Okay, so have you heard of Wnt signaling? Wnt signaling has the direct readout of beta catenin, as like the direct readout of Wnt signaling is beta catenin. So that we can say that Wnt is directly related to actin cytoskeleton reorganization as well as other functions. But this is something that we have to keep in mind as we go on, that beta catenins can have the effect on cell adhesion directly, and which is a downstream target of Wnt signaling. This is something that we will discuss in the next week's class as well. So, and this was already answered. Catenin stabilize cell adhesion by interacting with actin cytoskeleton. Let us continue. Which of the following molecules is or are responsible for apical constriction during ventral furrow formation in Drosophila embryos? The options are DPP, twist, rho GTPase, and actin. There is more than one type of correct option, so like more than one is correct. So anyone wants to try and answer this? Sneha says 2 and 3, twist and rho GTPase. Deepak also says 2 and 3, twist and rho GTPase. Ujjwal and Milani also says 2 and 3, twist and rho GTPase. Okay. So let us look at the particular solution slide. So this is the schematic representation of ventral furrow formation in Drosophila embryo, with left to right progressing as time progresses. So obviously the furrow forms on the ventral side, and we can see that the ventral cells are defined by expression of transcription factor, twist and snail. So only in the ventral cells are the transcription factor, twist and snail expressed. These cells accumulate myosin 2 at their apical surfaces. So for all else, we can see there is a basal localization of myosin 2, whereas only these cells have an apical localization of them. When myosin 2 interacts with actin already present, the cells begin to constrict apically and thus invaginate. So essentially this happens as such. We have seen money pouches that have strings on either end. So when you pull the string, the entire pouch closes, and something similar happens. And due to the apical constriction, the entire cell population moves inward. And this is a close up of the entire thing. where before initiation of the ventral furrow, rho green and beta catenin both reside along the basal surfaces of the ventral cells. Beta catenin is also formed in the subapical region, and this is what in turn allows for the apical constriction to occur. So the GS, like the transcription factor, the proteins associated with it all are twist, snail, myosin 2, rho, beta catenin. As I said, we have already have encountered beta catenin, and the actin that is already present inside the cells. So actin is present everywhere in the cells, but in these cells only does the interaction with myosin allow the actin to actually allow the actin to actually start the invagination process. Or in all the other cells, even the actin is present, myosin is not activated, and thus only these cells start invaginating, and nowhere else can we see the invagination occur. Thus, the correct options are twist, rho GTPase, and act. Anyone wants to add anything to the question? I guess not. Okay. We continue. Which of the following are true regarding invagination of cells during gastrulation? Invagination during Drosophila gastrulation occurs on the dorsal side. Formation of furrow begins with relocalization of rho and beta catenin. In Drosophila, the mesoderm is formed from epithelial cells on the ventral side of the embryo. In apical constriction, the apical constriction is responsible by the expression of twist, snail, and myosin 2 genes. So there was a typo here. It should be myosin 2, so I have added. It looks strange, but it is myosin 2. So which of the following are correct? Mriani says 2. The formation of furrow begins with the relocalization of rho and beta catenin, and also 4. Sneh also says both 2 and 4. Deepak says 2, 3, and 4, that in Drosophila, the mesoderm is formed from epithelial cells on the ventral side of the embryo. And Shiansh says 1, 2, and 3. Invagination during Drosophila gastrulation occurs on the dorsal side. Formation furrow begins with the relocalization of rho and beta catenin. And mesoderm is formed from epithelial cells on the ventral side of the embryo. J. Surya says 2 and 4, which is formation furrow begins with the relocalization of rho and beta catenin. Apical constriction is responsible by expression of twist, snail, and myosin 2 genes. Okay, so with our we'll go back to this particular slide. So with our last discussion, we know that the ventral furrow is called ventral furrow because it forms on the ventral side, not dorsal. So I think option 1 is incorrect. Probably that was the option that says invagination starts on the dorsal side. It does not. It starts on the ventral side. Another option, twist and snail localization, rho and beta catenin localization is what initiates. That is correct. That is what initiates the ventral furrow formation. And what was the other? Apical constriction is responsible for twist, snail, and myosin 2. This is also correct. In Drosophila, the mesoderm is formed from epithelial cells on the ventral side of the embryo. So we go back to our discussion on the various types of cells. Epithelial cells do not form the mesodermal layer because epithelial cells occur very much later. Mesodermal, the germ layers occur first. So there is nothing called epithelial cells in the beginning. Mesoderm does not form from the epithelial cells on the ventral side of the embryo. The essential reason is that when the cells migrate inside, they also will give rise to the endoderm as well. So the options are formation of furrow begins with the relocalization of rho and beta catenin, and apical constriction is responsible by epithelial expression of twist, snail, and myosin 2 genes. 2 and 4. Anyone has anything to add to this? Okay. Next, the function of Drosophila protein branchless is an example of rearrangement of actin polymerization leading to cell shape change, chemoattraction, external signal that influences cell shape change, and rearrangement of cadherins leading to cell shape change. This is more than one type of options are correct. Anyone wants to try and answer this? Deepak says 1, 3, and 4. Rearrangement of actin polymerization leading to cell shape change, and external signal that influences cell shape change, and rearrangement of cadherins leading to cell shape change. Milani says 1 and 4, and Jaisuri also says 1 and 3, which is external signal to influence cell shape change. I missed anyone? I do not think so. Okay, so let us look at what exactly branchless is. During trachea formation in Drosophila, there are particular cells known as branchless secreting cells. These cells secrete a particular chemoattractant are particular molecules that allow cells to migrate, that they are attracted towards the source of the chemical and they can start migrating towards it. And nearby cells secrete branchless protein, which activates breathless protein on tracheal cells. So these particular cells are known as leader cells, in which the breathless protein is activated in response to branchless protein secretion by these cells. And now these cells will try and migrate towards the secreting cells. The activated breathless protein induces migration of leader cells and tube formation. Thus, essentially, when the leader cells migrate towards the branchless secreting cells, we have a formation such as this, which later allows for branching structures to form in the leader cells as well. So these will also form the branching structures which actually form the trachea. So the function of Drosophila protein branchless is an example of chemoattraction, an external signal that influences cell shape change. So rearrangement of cadherin leading to cell shape change. So this is something that I would like you to understand is cell shape change is something that occurs when the actin cytoskeleton of the cell changes. Actin cytoskeleton is what controls the entire shape of the cell. Cadherins control what cells, what kind of external surface to which the cell can attach. Both are different things, and in between of them both lies catenin molecules which help the interaction between cadherins and the actin cytoskeleton. Thus, branchless is an example of chemoattraction, an external signal that influences cell shape change. Anyone wants to add something to this?
So,
Yes, Deepak, go ahead.
so during the lecture, it was said that internal change in the cell affects the cadherin that is present on the membrane, right? But sir also mentioned the vice versa is also true. So in that regard, can the fourth option be sensible in any form?
Okay, so rearrangement of cadherins leading to cell shape change. Okay, that might be correct. I would have to look into it. But as far as I think, the B and C are correct that I know. D I will have to look into and we'll get back to you. Okay.
Okay, sir.
Okay. So we go on to the next question. During the initiation of cell migration, polarization signals initiate. The options are the interaction between Rho GTPases and WASP-N, binding of myosin to actin, wide-scale reorganization of the actin cytoskeleton, and the link between actin and integrin. So now here we have moved on and now discussing cell migration. So during cell migration, when a particular cell decides that it has to move to a separate location, various steps are involved. The one of the primary steps is the polarization of the cells, in which the cells understand that one of its ends needs to move, and in that particular and in that moment it also recognizes the direction it has to move to, and that is what is known as polarization. So during the initiation of cell migration, the polarization signals initiate which of the following first: the interaction between Rho GTPases and WASP-N, binding of myosin to actin, wide-scale reorganization of actin cytoskeleton, and the link between actin and integrin. This is one option correct answer. And okay, Deepak says number 3, that is wide-scale reorganization of actin cytoskeleton. Anyone else wants to try? Okay. So let us look closely into what exactly are the steps in cell migration. So here we can see a migrating cells with migrating cell which first looks like this, and then for when we stain with uh, with a particular stain with the particular antibody which binds to beta catenin, we see redistribution of the initial cell and how it changes. So blue shows us the filamentous actin that is being formed, and there is a gradient. So the blue is the highest gradient of actin polymerization, filamentous actin, followed by green. So we see in this edge of the cell there is a huge concentration of filamentous actin present. So in the laminopodia migrating mesenchymal cell, the ratio of filamentous actin to globular actin is visualized by different colors. Blue being the highest level filamentous actin level and red being the lowest. So we can see there is a particular polarization in which filamentous actin is formed in the laminopodia region. So let us look into what exactly the other steps of migration. The first stage of migration is polarization, wherein the cell defines its front and back. So this is the front part and this is the back part. Polarization can be directed by diffusing proteins such as chemotactic protein or by signals from the external matrix. As we have just seen, the branchless protein was one of them, in which allowed particular polarization and then migration. The second stage of migration is protrusion of the cell's leading edge. The mechanical force for this is the polymerization of actin microfilaments at the cell membrane, creating long parallel bundles which are known as filopodia, or broad sheets such as here, which is known as laminopodia. Third stage is migration involving adhesion of the cell to the extracellular substrate, and fourth concerns the release of adhesions which were on the back side of the cell. So first it attaches to ECM that is present here on the in the direction it is moving, and then deattaches from the ECM that it was attached to in the previous position. This is the entire flow chart of how exactly the migration occurs. So first polarization, followed by protrusion of one edge, which can be in the form of filopodia or laminopodia, which is done by actin polymerization, followed by adhesion to ECM in the front part and detachment in the previous part. So if we go back to our question, the polymerization signals initiate. So anyone will now like to try and answer the questions after the discussion itself? Anyone else will try other than Deepak? Rani says 3, Sneha also says 3. Yes. Okay, so I guess everyone else also understood that it will be 3. Wide scale reorganization of the actin cytoskeleton is what the polarization signals initiate. Sangeeta also says 3. Okay. So we move on. Which of the following statements are false with regards to cell migration? Less filamentous actin is present on the moving side. Cell migration happens primarily due to reorganization of the actin cytoskeleton. Addition to the ECM integrin happens before polarization of the cells. Myosin and its regulators provide motive force along the actin microfilaments. So, using what concepts we have just learned in the previous discussion, which of these are false? So melanosis 1 and 3, less filamentous actin is present on the moving side. Addition to the ECM integrin happens before polarization of the cells. Deepak says 4 is also correct. Myosin and its regulators provide motive force along the actin microfilaments. Sneha also says 3 and 4. Myosin and its regulators provide motive force. And addition to the ECM integrin happens before polarization of the cells is false. Jay Suri also says the same. So the false options are 1 and 3. That is, less filamentous actin is present on the moving side, as we have just seen here in this diagram. More filamentous actin is present on the moving side. This is the moving side. That is where the laminopodia is there. And here we have not talked about what exactly creates the motive force for moving, but that is what the action of myosin and its regulators are. They provide the motive force. So what exactly is pulling the cell towards the chemoattractant region? That is the myosin. And the other options, so these two options are correct. Addition to the ECM integrin happens before polarization of the cells is obviously false, because as I said, ECM first the cells attach to the ECM in the first there is polarization, then there is movement, then there is ECM addition, and then there is ECM release. So polarization happens way before ECM integrin addition happens. Is that okay? I guess. Okay, so we move on. The next question is, normally optical vesicle in the developing frog embryo induces lens formation only in the anterior surface ectoderm. However, in a newly generated mutant, it induces lens formation in the other regions of the ectoderm as well. What do you think is happening in the new mutant? BMP4 is not produced in the mutant optical vesicle. Competent factors is not produced by the mutant optical vesicle. BMP4 is expressed in the entire surface ectoderm of the mutant, and PAC6 is expressed in the entire surface ectoderm of the mutant. So this is one option correct. If anyone wants to try, they can. Or we can. Okay, Sneha goes right off and says PAC6 is expressed in the entire surface ectoderm of the mutant. Anyone else? Okay, let us first go through the solution slide and then try it. So this particular question touches upon the concepts of induction and competence. Induction is a particular term in which we define that a particular tissue can produce certain signals which induces another set of tissues to form a specific structure. Competence is the term which defines that the tissue which is receiving the inducing signal can change accordingly, can respond to the inducing signal. These are two separate concepts. So you can essentially, for a very simplified way in which we can explain this, it can be said that it is like a jigsaw puzzle. In a jigsaw puzzle, the protruding end which inserts into the socket of another jigsaw puzzle. So this, if it is the protruding end and this is the socket in which it will go, this can be said as the inducing signal, and the formation of the shape of the socket is the competence. So without the inducing signal and the competence being there, both being there, there can be no formation of the resulting tissue. So this is something that is explained by this particular diagram. So we have what here we have the optic vesicle, which is a structure that forms by development, and this optic vesicle signals to the anterior epidermis of right here to so this optic vesicle induces the anterior portion of the ectoderm right here to form the lens. Thus the optic vesicle is inducing the ectoderm in order to form the lens, and the lens ectoderm should be competent to receive the inducing signal and form the lens by the optic vesicle. So the ectodermal competence and the ability to respond to optic vesicle inducer in Xenopus. The optic vesicle is able to induce lens formation in the anterior portion of the ectoderm But so optic vesicle is present and competent ectoderm is present, and then only we see lens formation. So this protrusion right here. But say the optic vesicle is present, that is the inducer is present, but it is somewhere down here where the cells are not competent, and thus we see no lens formation. Here the optic vesicle is not present, but competent cells are present. But here also we can see no lens formation, because obviously there is no inducing signal. And here we have a another tissue which is not optic vesicle and but competent cells, and thus there is again no lens formation because the inducing signal is absent again. So this entire diagram tells us that both the inducing effect of one tissue and the competent cells need to be present in order to form particular structures. So if that is okay, we can go back to so I will give you another piece of information, that the master regulator, so Deepak is already answering, we go back here. So the master regulator of lens formation is PAC6. PAC6 is the competence factor that is present in these ectodermal cells. So with that information, let us try and answer the question again. What the question is saying is that developing frog embryo induces lens formation only in the anterior surface ectoderm, just as we have seen. However, in a newly generated mutant, it induces lens formation in other regions of the surface ectoderm as well. So, if the condition is something like number 2 here, so optic vesicle, wherever you put it, there will be lens formation. What do you think is happening in the new mutant? Now look at the 4 options and try and answer this. BMP4 is not produced in the mutant optical vesicle. Competence factor is not produced in the mutant optical vesicle. BMP4 is expressed in the entire surface ectoderm of the mutant, and PAC6 is expressed in the entire surface ectoderm of the mutant. So I would request, please try and answer this. So Deepak changes his answer to 4, which is very nice. PAC6 is expressed in the entire surface ectoderm of the mutant. Anyone else wants to try? No, I guess not. So a lot. Yes, this is the, so the option is PAC6 is expressed in the entire surface ectoderm of the mutant. Why do I say so? As I said, PAC6 is the competence factor. That is, due to the presence of PAC6 in these ectoderm cells, they were able to be induced to form the lens. So whichever cells has PAC6 or the competence factor, after receiving the inducing signal, can form the various structures. So in the new mutant, obviously the PAC6 was expressed in the entire ectoderm, and thus wherever I replace the optic vesicle, there the lens was being formed. Thus the answer is 4. PAC6 is expressed in the entire surface ectoderm of the mutant. Is that clear or no? Please tell me, because this is a very important thing. The, like, one of the basic concepts of developmental biology, the induction and competence, and I would like it to be very clear. Do you have any questions regarding this? Okay. So we move on and look at the following. What will be the most likely effect on development if the foot dermal mesenchyme of chick is placed in contact with quail wing epidermal epithelium? So we have seen these experiments being discussed in the lecture, in which if we am I is the screen visible? Hello, am I audible at all? Okay. So okay. So the question is, what will be most likely effect on development if the foot dermal mesenchyme of chick is placed in contact with quail wing epidermal epithelium. Formation of quail scales and claw. The epithelium will degenerate due to genetic incompatibility. Formation of chick scales and claw. Formation of chick wing feather. So this is one of the things that was discussed in the lecture. The epithelium that is present. So the various experiments were done in which epithelium from various parts of the chick was mixed with mesoderm from various parts of the chick as well. So when this was done, it was observed that irrelevant of where the epithelium is from, the defining characteristics of the epithelium, say formation of thigh feathers, formation of wing feathers, or formation of the scales, is determined where the mesenchyme is taken from. So if the mesenchyme is taken from the wing part and any epithelium is kept on top of it, it will form wing feathers. Similarly, mesenchyme taken from the feet part, any epithelium is put on it, it will form the scales and the claws. Like that is the characteristics of the foot part. So the mesodermal origin or the foot dermal mesenchyme is what essentially describes what the characteristic of the epithelium will be. So a separate experiment is done in which foot dermal mesenchyme of the chick is placed in contact with the quail wing epidermal epithelium. So a cross species experiment. So with the information that the mesenchyme will direct what kind of structures will form, what do you think will the quail wing epidermal epithelium, once it is allowed to develop, what will its characteristic be? Will it form quail scales and claw? Epithelium will degenerate. Formation of chick scales and claw and chick wing feather. Anyone wants to try and answer this? Ujjwal, Sneha and Deepak, all of them say 3. Formation of chick scales and claw. Anyone else wants to try? Okay. So let us look at this another cross species experiment and try and correlate. Here, to the donor and the host, as I said in the question, the donor and the host are quail and chick. Here, the two were frog and newt, which are also very closely related species. When the frog's area of presumptive oral ectoderm, that is, this particular area will form the oral ectoderm part, is placed in the newt gastrula, we can see newt with frog tadpole suckers. That is, the newt develop tadpole-like features. Similarly, when the opposite was done, the frog developed newt-like features. So this tells us about the genetic specificity of induction. That is, as the previous experiment we described, mesenchyme directs the formation of the epithelial epidermal characteristic. So wing mesenchyme, any epithelium you put, it will form wing-like characteristic. Feet mesenchyme, you put any epithelial cells over it, it will form feet-like characteristic. But when you do cross-species experiments, what happens is mesenchyme goes ahead and says form wing-like characteristics. But the epidermal cells, they know only to form, say in this particular example, when the chick mesenchyme is placed, when the quail wing epidermal epithelium is placed on top of chick mesenchyme, the chick mesenchyme says form feet-like characteristics. But quail does not know how to, quail epidermal cells do not know how to form chick scales and claws. They only know how to form quail scales and claws. And thus, when the mesenchyme signal is there that to form foot dermal like feet-like characteristics, they will go ahead and form quail scale and claws instead of chick ones, because they do not have the information to form the chick scales and claws. The only signal coming from the mesenchyme is not the entire signal go ahead and form this particular chick scales and claws. No, the signal that comes from the mesenchyme is form feet-like characteristics, and then the epidermal layer will now interpret it and then form the particular feet-like characteristics that it knows. Since the quail wing epidermal cells were taken, they will go ahead and form quail scales and claws when placed over feet dermal mesenchyme of the chick. So the answer is number 1. So this was very confusing. I go ahead and explain again if anyone has any confusion regarding this.
Yes, I would like one more explanation.
So okay. So okay, we can do this. My screen is still visible, right?
So,
Let me go to the experiment itself. I would actually request everyone to try and read the book because it has a lot of material that you would find helpful. So this is the particular experiment we are discussing, in which we take a particular mesenchyme. So my cursor is visible, I guess. So we take a particular dermal mesenchyme and we put wing epidermal epithelium on top of it. Depending on where the dermal mesenchyme is from, irrelevant of where the epithelium is from, the mesenchyme will direct formation of specific structures from where the mesenchyme is from. So if we take mesenchyme from the wing, it will form wing feathers. If we take mesenchyme from the thighs, it will form the thigh feathers. If we take mesenchyme from the foot, it will form scales and claws like the foot, irrelevant of the way the epithelium is formed. So in order to understand exactly what is happening, so what is the signal coming from the mesenchyme? People did go ahead and did this kind of cross species experiments. What they did is exactly similar to what was done in the previous experiment. They took area of presumptive oral ectoderm and placed it over. So essentially, they took the skin epidermal layer from one donor, which is the frog, and placed it over the mesenchyme of the newt mesenchyme and frog epidermis. And now they tried to observe what exactly is being formed. So as they expected, mesenchyme, because the mesenchyme is obviously in the oral region, the mesenchyme told the new epidermis to form oral structures. But since the tadpole only had the information to form tadpole-like oral structures, which are the tadpole suckers, only the frog tadpole suckers formed instead of newt balancers. So in newt, this particular portion will form balancers, and in frogs, this particular portion will form suckers. So what this particular, and reverse is also true, when the newt epidermis is placed over the frog mesenchyme, the mesenchyme commands the epidermis to form oral structures, but the epidermis only knows to form frog-like oral structures, and thus we will see these balancers for newt-like oral structures, and thus we will see this balancer forming. So this just tells us that mesenchyme, though the signal from the mesenchyme is, it is just asking the epidermis to form specific structures that are characteristics of the mesenchyme. So wing mesenchyme will form wing structures, feet mesenchyme will form feet structures. But epidermis, it is up to the information of the epidermis to form what exactly the feet structures look like. So in our question, the the foot dermal mesenchyme of the chick commands the quail wing epidermis to form feet-like structures. Quail wing epidermis says, okay, feet-like structures, but it only knows how to form quail feet-like structures, and thus it forms quail scales and claws. So the mesenchymal signal is only that form feet-like structures, not what exactly you have to form. You have to form feet-like structures. And epidermis decides based on what the genetic specificity of the epidermis is, it will form the specific feet-like structures from where the donor is. So quail epidermis will only form quail scales and claws. Is what exactly the entire question entails. So this is a very long discussion which essentially says that foot dermal mesenchyme of the chick will ask the quail wing epidermis cells to form feet-like structures, and quail wing epidermis cells will form feet-like structures, but only of the quail characteristics, and thus we have formation of quail scales and claws. Is that clear?
But sir, isn't in the book it is said that mesenchymal cells will direct the development?
Yes, yes. So mesenchymal cells direct the epidermis to form feet-like structures. See the mesenchyme here is foot mesenchyme from the chick, and the epidermal cells are from the epidermis is from the wing. So mesenchyme forces the epidermis to have feet-like structure. That is, the mesenchyme is directing formation of structures in the resulting chimera. But since the mesenchyme signal is only forming feet-like structures, quail wing epidermis will form the structures that feet-like structures that it knows, and quail wing epidermis only knows how to form quail scales and claw, and that is why we will have final formation of quail scales and claw here. Is that okay or no?
So, if I'm understanding it right, for some reason the epidermis is because the epidermal cells do not know to form the chick scales and claws. Then, in that condition, even after the signal from mesenchymal cells of chick, due to their lack of signaling or information, they are following the usual quail scales and claws.
Yes, exactly. That is the essential point. The quail wing epidermis does not know how to form the chick scales and claws. When the signal comes from the foot dermal mesenchyme of the chick to form feet-like structures, the quail epidermis forms the feet-like structures that it knows, and it knows how to form quail scales and claws, and that is why it will give rise to this structure. This is essentially what this experiment also entails. The when given signal from the newt mesenchyme to form oral structures, the frog epidermis will go ahead and form oral structures. But since it knows only to form frog-like tadpole suckers instead of the newt balancers, we will have tadpole suckers on top of the newt mesenchyme. That is essentially entirely what the question entails and what this particular experiment entails as well. I will request everyone to just go ahead and go through this particular experiment if you are still confused. But if you are still confused, you can go through it again. Anyone wants to ask anything else?
So here what is happening is the type of organ will be determined by the mesenchyme, but the species to which that organ will be similar or the origin will be determined by the epidermal epithelium, right?
Yes, exactly. The origin of the epidermal guides what kind of what the organ specifically will be, but the mesenchyme guides what organ it will be. Yes, that is also correct. Yes, exactly. So with that, I guess we can move on. So the next question is, how does the RTK pathway mediate its effect on target cells? So RTK, receptor tyrosine kinase, this was the final topic that was discussed in the lectures. And how does it mediate its effect on target cells? The options are reorganization of the cytoskeleton, post-translational modifications of target proteins, chromatin modification, and transcriptional regulation. So anyone wants to try and answer this? Okay. Ujjwal says 4, transcriptional regulation. Deepak says 1, reorganization of the cytoskeleton. Mrinalini says 4, transcriptional regulation. And Shriyan says B, that is post-translational modification of target proteins. So let us go through the receptor tyrosine kinase pathway and try and answer this. So this is a schematic showing us the receptor tyrosine kinase pathway. The pathway essentially starts with a ligand which binds to the receptor, following by dimerization of the receptor and autophosphorylation of the receptor. So autophosphorylation is there. So if we have phosphate group attached to the various parts of the receptor, Then, so the ligand activates the RTK by phosphorylation, followed by GNRP, RAS, RAF, MEK, ERK, and finally, ERK is essentially binds to an inactive transcription factor and activates it. And this action, now the active transcription factor present in the nucleus can go and modulate transcription. So essentially, the entire readout of the RTK pathway is transcriptional regulation in the cell that is receiving the ligand. So the entire pathway will end up in regulation of transcription. So the RTK pathway mediates its effect on the target cells via transcriptional regulation. Is that okay? I guess it is. So that is all we had to discuss today. The references for the slides are Introduction to Developmental Biology, Scott Gilbert, the 9th edition. And this is the particular YouTube link where all the videos are uploaded. This is also now available on the NPTEL site itself. If so, if anyone has any questions, you can go ahead right now, or we can end the session. Okay. Thank you everyone for joining the session. I look forward to having another session next week. Thank you.